secretion of proteins. When our desired protein it is expressed, then it may have biotechnological application. So under such situations, it must be released from the cell into the external environment, from where it can be collected for further use. So for this purpose, microbes must have efficient transportation system so that when protein it is being synthesized within the cytoplasm, then it must be efficiently transported across the inner or outer membrane of microbes. So host organism, as I quoted number of times that, for example, if it is E. coli, it is a type of gram-negative bacteria. If we have a look on its structure, then this host organism, it contains two membrane system. First membrane, it is called as inner membrane or cytoplasmic membrane, and after the cytoplasmic membrane, on outer side a cell wall is present, and then external to the cell wall an additional membrane is present, which is called as outer membrane. And the space between outer and inner membrane, it is called as periplasmic space. Now the expressed proteins, it can be transported from the cytoplasm to the periplasm, and then from the periplasmic space to outside the cell. And here different proteins, they again can assist in the process of transportation that we are going to discuss later on. At this moment, we can say that the universal mechanism that is important for the export of expressed proteins, it is called as general export pathway or GEP. And in general export pathway, there is assistance of different type of facilitating proteins or helping proteins, which simply can be abbreviated as SEC or SEC proteins or secretion proteins. Now during the process of GEP or general export pathway, proteins that are the substrate or target for export, they have a specific N-terminal signal sequence. This N-terminal signal sequence, it facilitates the movement of proteins from the cytoplasm to the external site. अगर हम ये देखें N-terminal signal sequence को, तो इसके हमें तीन मेजर डोमेन्स या पार्ट्स नजर आएंगे. इसका जो आउटर पार्ट है, N-terminus जिसे कहते हैं, it is positively charged. इसके बाद जो central domain है, it is hydrophobic in nature, so it contains most of the amino acids that are hydrophobic. And after that, there is a cleavage site for peptidase, because when proteins they are exported, their N-terminal signal sequence, it is cleaved with the help of enzyme, which is called as peptidase. So these are the three domain of N-terminal signal sequence, and overall we can say that कि ये जो N-terminal signal sequence है, ये प्रोटीन की ट्रांसपोर्टेशन में हेल्प करता है, स्पेशली उन प्रोटीन को जो कि साइटोप्लाज्म से बाहर की तरफ मूव करती हैं. ये तो सिचुएशन हो गई ऐसी प्रोटीन्स की जो कि नेटिव हैं, सिंगल एक्सप्रेस होती हैं. जैसे मैंने कहा कि बाज़ दफ़ा प्रोटीन्स अगर फ्यूज्ड फॉर्म में एक्सप्रेस हों, जिसको हम हाइब्रिड प्रोटीन्स कहते हैं या फ्यूजन प्रोटीन्स कहते हैं, तो इनकी स्टेबिलिटी कंपैरेटिवली ज़्यादा होती है. तो इसके लिए जो सिस्टम है ट्रांसपोर्टेशन के, वो इतने वेल नोन तो नहीं है, लेकिन ये रिपोर्ट किया गया है कि सच प्रोटीन्स में हैव बीन ट्रांसपोर्टेड even in folded form or precipitated forms. So in this case again, different type of SEC proteins or SE proteins, they are involved in this process. The export of fusion protein, it was first time reported around 1978 when workers were able to report the transportation of such fusion proteins like the proinsulin from the rat along with some beta-lactamase enzyme. So such fusion proteins, they were exported successfully in case of E. coli. In addition to that, the transportation of the proteins across the cytoplasmic membrane, it may be interrupted by different type of proteases that are present. And so that especially when proteins, they are present in unfolded state. When they are in unfolded state, lack of tertiary structure, then proteases can target them and degrade them. So to avoid this, there are certain families of chaperone proteins within the periplasmic space, like DSBA protein or chaperone. So this DSBA chaperone protein, they can introduce disulfide bonds in target protein like alkaline phosphatase, suppose, so that they can be prevented from degradation. Otherwise, if such chaperone proteins are not there, like DSBA chaperone protein, so alkaline phosphatase, it will fold very slowly, and even if protease target them, then they can also be degraded. So for the export of proteins, as I told you that the main system, again we can say that the overall it is a general secretory pathway or GSP that we are going to target in next topic. And in this pathway, different set of proteins, they are facilitating the movement of proteins across the membranes of microbes.